During recent years, the amount of biological data has significantly increased. Many medical improvements have been achieved and many diseases tackled using structured data analysis and data science methods involving biological data. Large amounts of this data are available online for public use. Some people and organizations already speak about the fourth industrial revolution, addressing the expected impact of bioinformatics on medical, biological, and healthcare advancements. A large amount of open data, many unanswered questions, and high impact of research sounds intriguing for data scientists like myself or other data experts like data analysts, data engineers, software developers, or students. But when you try to get inside the field, you find out that it is very complicated and messy. The first thing that comes in our way is the understanding of biology. To successfully manipulate the data and draw conclusions, one should have some understanding of the underlying biological processes. These processes are often complex and require lots of studying. The second thing is software. Researchers have been developing independently their own analysis software for their research questions, which led to the situation where we have many different independent pieces of software. There are no uniform APIs or frameworks for data manipulation and analysis like there is for traditional data science. The first steps towards the standardization and integration have already been done, but there is still very much to do. For the same reasons, we have the third problem: data formats. They vary a lot. There are hundreds of formats for different research applications. This is why self-studying of bioinformatics may feel like a very complicated puzzle. The purpose of this series is to introduce people from different areas of IT to the bioinformatics research field, keeping the focus on the application side, explaining only the relevant part of underlying biological processes. With this approach, professionals and students from data and software areas can understand the ongoing development, make their own bioinformatics projects, join open source projects, or start their career in bioinformatics. The topic is very large and I tried to create a systematic approach where we can start from simple things, moving deeper with each video. We begin our journey with DNA, as it is a basic but very important concept and some may even remember it from high school. We will look at how the DNA information is stored and manipulated on computers and try it ourselves. But before doing DNA computations, we need to familiarize ourselves with the necessary basic knowledge about DNA. Our organism and the organisms of all living creatures consist of cells. Some organisms have only one cell, but mainly cells act like building blocks of an organism. Cells are able to do many things, including replicating, which means creating copies of themselves. To make this happen, a cell should store information on how to build itself. And this is exactly the role of DNA. Broadly speaking, the DNA is the recipe for the different structural components of the cell. Without the DNA, we would have only a bunch of unrelated chemical structures. When the cell replicates, the DNA is also copied to the children cells, and the cell replicating process can iterate over and over again. Every living creature on the Earth stores its biological information in the DNA. In a single creature, every cell contains the same DNA. This means that, for example, any cell from your eye, heart, or skin has exactly the same building information. While having the same building recipe, the cells are obviously different. While DNA transfers all the building information, there are other biological processes that decide what parts of DNA to read and use when building the cell. This is a very interesting research area and we will get there in the future. So now you know that the main function of DNA is to carry and transfer biological information. But how exactly does this happen? We can compare it to computer memory. Computer stores information in the form of zeros and ones. To store this physically, it is encoded as a charge on and charge off. Of course, light bulbs are not used nowadays for this purpose, but you get the idea. Instead of ones and zeros, information in DNA is represented with 4 letters: A, T, G, and C. Physically, they are represented with 4 different chemical structures called nucleotides. The letters A, T, C, and G come from the names of these structures. The DNA is just a long strand created from these 4 chemical structures. And on the informational level, each structure is represented with 1 letter. So from a computer science point of view, DNA is a string that contains only the letters A, T, C, and G. This string is typically very long. For example, the DNA carried by one human cell has approximately 6.4 billion letters. Now you may see why we do need data science here. To store and transfer the DNA strings, there must be some file format. There actually exist many different formats to store DNA sequences. Luckily, there are some generally used formats that are getting close to becoming conventions. The easiest way is obviously to just copy and paste the string to a text file and save it as a TXT format. This is called plain sequence format. The drawback here is that it is not possible to store any metadata, IDs or description. The general formula for DNA storage format is sequence plus metadata. The sequence is the string containing DNA, and metadata is any data that is decided necessary to store with the DNA, like for example the origin of the data. Probably the easiest and most used way to store the DNA sequences is the FASTA format. FASTA format may have extensions like for example .fasta, .fna or .ffn. The extension doesn't really matter, but it may help you to identify faster what file it is. The file starts with a greater than character and any metadata. Starting on the next line is the DNA sequence. Here is an actual example of a FASTA file. FASTA may include multiple sequences, and each sequence will have 1 line for metadata. For comparison, let's look at another 2 formats, EBL and GENBANK. If you want to look closer, you may pause the video. As you see here, the structure is the same: sequence and metadata. You don't need to know the further structure of these formats, how to read the metadata or how many spaces and tabs each format has. The software will do the reading and writing for you. You should just know that these file formats carry the DNA. Other similar formats are GCG, EG and FASTQ, although FASTQ is usually used for a little different purpose. Now we can move on to actually work with the DNA data. Many different programs exist for manipulating DNA data. I will use Python here as I think that it is a step towards the integration to other data science disciplines. The package called Biopython is an open source project especially developed to create easily accessible libraries and applications for bioinformatics. All the code shown here is available in GitHub and the link is in the description. Biopython is available from pip using pip install biopython. To work with sequences, there is a module called sec. It encapsulates the string object and also provides some specific functions. Usually you never write DNA sequence by hand and load it from a file. For educational purposes, let's create one manually. First we import the module. and then create a sequence. The letters in sequence can be accessed by index. Here is the first letter and here is the second. It is also possible to slice the sequence. Let's select a part of the sequence. There are also many other functions, but at this point it is enough for us to know these ones. Now let's open one real DNA file and make our first example with real data. For this example, we look into the DNA of a bacteria that causes tuberculosis. Its official name is Mycobacterium tuberculosis. As I said before, biological data is available online on different web resources. One main website is NCBI, National Center for Biotechnology Information. The link is given in the description. NCBI is a huge portal with many different databases, and we will get more familiar with it in the future. Now we type the name of our bacteria in the search field and select nucleotide from the drop-down panel on the left. Nucleotide is an international database of public DNA files obtained from organizations and researchers, and it is updated on a daily basis. DNA files are usually related to research papers or patents. It has a huge amount of different DNA files, so you need to really know what you are searching for. Our search produced many results, and I will select the second instance, which is one full genome of Mycobacterium tuberculosis. Genome is the full biological information carried by one organism, meaning it's all the DNA information that this organism has. Here the system shows me the GENBANK instance. To download the file, I click Send to in the upper right corner, click File and select the format. Here I choose FASTA and click Create File. We could as well download it in GenBank format, it doesn't really matter. Biopython can open it as well. Now we get back to our script. To open this file, we import sec.io, sequence input output, a module for reading and writing sequence files. The reading part of the code looks like this. I have to specify the file name and the format. Because file can contain multiple sequences, we iterate over the reads and save them to array. Our file contains only one sequence, so we save the first element of the array to the variable tuberculosis. If I now try to print the sequence, I will be able to see it only in limited format, because the sequence is obviously too long to print it on the screen. We can actually also get the length of the sequence. As our first data analysis example, we will plot the counts of different nucleotides as a bar plot. This is rather simple code if you are familiar with plotting data. As a result, we get the following plot. We counted how many letters T, G, C and A are in the genome of Mycobacterium tuberculosis. Each bar represents one type of nucleotide, and on y-axis we have the total count. Here we can make our first bioinformatical conclusion. The amount of G nucleotides is approximately same as C. The amount of T's is approximately same as A's. This is not a coincidence, but to explain that phenomena we will have to study a little more. Now let's answer the simpler but very central question of DNA analysis. If DNA is always copied during replication, how it is possible that different organisms have different DNA? In fact, DNA is not always copied exactly. Replication is a complicated biological, chemical and physical process, and sometimes errors do occur. These errors are called mutations. You have probably heard of mutations before, but what does it really mean? Assume we have an imaginary cell with this piece of DNA. If the DNA is copied correctly during replication, the newly created cell will have an exactly same DNA. Consider the following case. The copying process goes as intended until this point. Here should be G, but because of a copying error it becomes A. This is called substitution, but the word replacement is probably easier to remember. Consider now another one child of the cell. The replication again starts normally, but here, where it should be G, an error happens and it is never added. The process continues directly to C. This situation is called deletion, and it happens when one letter from an original DNA is dropped out during replication. And finally, in the last example, the process reaches G and it suddenly puts there two unrelated letters. Then it continues normally from the G. This one is insertion. Insertion, deletion, and replacement are three mutation types. They may be further divided into subcategories, but this is good enough for us now. Mutation can lead to medical diseases, physical differences, variety of species, evolution, and many other important processes. To computationally analyze all these, we should be able to compare different sequences. But how to do it when the sequences have mutations, can have different lengths, and letter combinations may be the same but in different positions? It is an extremely important and interesting question that already has many solutions in production, and it will be the topic of the next video. The references are in the description, and if you have any feedback, positive or negative, please add it to the comment section. See you soon.